Oh, yeah. It's working.
Perfecto, perfecto.
You can hear. Thank you.
No. You
Está mudo.
Okay, now you can see my screen.
Sí, sí, perfecto.
Oh, please.
Yeah.
Okay.
Thank you.
And if I pass, they pass.
Yeah.
Sí, perfecto.
Good. So now you are seeing this. Okay, this is not optimal. Let me check. So yeah. So is there a way to see the screen that I'm sharing in addition to you?
I'm afraid it's not possible. So
Okay, good. Okay, just I'm very used to Zoom, so
Yeah.
but that's okay. That's okay. That's perfect. Okay, no problem. Okay, so now you are seeing my presentation.
Yes, we are.
Good. So I think that we should wait a few more minutes, right, Igor?
Exactly. We waited like 5 minutes maximum because then most of the students are already present.
Excellent. Okay, so let me go back to here. So how many students are typically in the course?
Normally we have around 20. Depends on the year. This time we have 19 students.
Nice.
Yeah.
It's good. That's good. People want to study biology. That's good.
Yeah. I mean, we were offering this course every year. There were some times that we have like more than 30 students enrolled. And then we observed that then the number of students reduced quite a lot because, I mean, they're doing their PhD, they will do this once and then they cannot do it again. So then we are offering every 2 years now.
Absolutely. Yeah. And this is something that is mandatory to the students to do courses. So this is
Yeah.
one option, or
No, we have no mandatory courses. They have
But
to get some credits, 14 credits. So they will choose the courses that
yeah.
are more appropriated.
It
I
is
see.
mandatory to do the courses, but there's no course that is mandatory.
I see. It's mandatory. Yeah. I see. So you need to do a given number of courses
Exactly.
to get the. Yeah, I see. Yeah, that's
And
perfect.
depending on the program, like in Botany, that this is our program, it's only like 14 credits that we say, which is easy for them to get. But some other post-graduation courses, they require like 30 hours, or I mean, it's really a lot.
I see.
It's
No, that's
similar
perfect.
as the one we have done in Buenos Aires.
Yeah, I don't remember now if I have to do courses at that time.
Yeah. You have done some
Yeah,
courses to get some credit.
probably.
Yeah.
So here, indeed, here. Well, the seminar that I will give today is mostly scientific. Right? It's not. But I typically give a lot of this type of courses but typically for master students, right? Because I don't know if you have a master degree. I see.
Yes.
And here, the PhD is very short, 3 years only. Yeah, so they need to rush. So they typically do the master. They do the master 1, master 2, so it's 2 years, and then they typically continue the PhD in the same lab where they do the master 2. So then they have a kind of a 4 years. And they have courses, but the courses are completely, in the PhD, they have courses that are these are mandatory, ethics in science, or finding your way after finishing your PhD. It's more of this type.
Yeah, 3
This
years.
is good.
Is
It's
really
good
short.
because you give the opportunity to them to see different things, different people.
Yeah, absolutely.
But sometimes the master degree is almost as with licenciatura, right? Because
Exactly. Yeah, we have, and particularly in Argentina, is so old-fashioned. The program is so old-fashioned that I mean, it's very difficult to match with what's happening in the United States or Europe. And indeed, for 5 years or more. Sorry, what is mute?
Magda, Leandro, I think I will start.
okay.
Wait, because my neighbor is like drilling the wall here. So, oh, it's okay now. So I will start. So good afternoon, everyone, the students that are here in the Google Meet, and everybody that is following us on YouTube. Today is a pleasure to introduce you Dr. Leandro Quadrana from the University of Paris-Saclay. Leandro, he will introduce himself, all his background. So Leandro, on behalf of our Department of Botany from the University of São Paulo, on behalf of Magda, Leandro, and myself, we thank you very much for your contribution to our course, and welcome, and thanks a lot for your seminar.
Okay, thank you, Igor. So you hear me well. First, I would like to thank Magda for bringing me here. Yes, both things. I will go to the special acknowledgement to Magda, but yes, to invite me to give this seminar. And you, Igor and Leandro, for this organization. It's very nice. It's very nice also to across the distance, share what we are doing in our lab. And a special thanks to Magdalena because, and in this way I also introduce myself. I did my PhD in Buenos Aires and in the laboratory of Fernando Carrari, which was a very strong colleague of Magdalena Rossi. So it happened to me that during my PhD, I interact a lot with Magda. We have done several projects together. I have visited her lab several times. We have spent days and nights doing qPCR. She was a great mentor. She always she has a very contagious energy, and this has continue with me till today. So thank you, Magda. Thank you, Magda, for all this support and help that you give me in these critical moments. And continue with the introduction is ideas. I did my PhD in Buenos Aires, working on tomato metabolism. And at the end of my PhD, I was trying to find a locus, a QTL that was underlying a vitamin E trait. And turned out that this was associated with a gene that contained a transposon element insertion in their promoter. And then I got obsessed with these transposon elements and their epigenetic regulation. And then I moved to France, to the laboratory of Vincent Collot, where I did a postdoc for several years. Continue working on to try to understand what is the contribution of transposon element, which is what I'm doing till now. After 4 years of postdoc, I got recruited as a permanent researcher in the CNRS, which is the national organization of researchers in France, the Conseil National de Recherche Scientifique. And since 3 years ago, I opened my lab in the University of Paris-Saclay, which is nearby Paris, right? Outside Paris. And in the lab, we are very much interested to understand what are the contribution of transposon elements, mobilization and their regulation to intraspecific diversity. And I hope that you are all related or know what a transposon element is. I will anyway introduce you. But one key aspect that you probably already face is that transposons are everywhere in eukaryotes. And this is, okay, let me put my pointer. Sorry. Okay, so they are very common in eukaryotic genomes, and this ranging from around 20% in the model plant Arabidopsis thaliana till almost half of the human genomes, which I like to say that we humans are sometimes more transposons than humans themselves. Maize, where transposon was first discovered, they're made up almost 90% of the genomes only made of transposons. The second thing that you can see in this plot is that the type of transposon that colonize the different type of genomes vary quite a lot. For example, in plants, they typically are populated by retrotransposon, LTR retrotransposon, you can see here in blue, whereas in mammals, they're typically enriched in this type of line elements, which are non-LTR retrotransposons. Fishes, on the other side, are mostly composed of DNA transposon. And we still don't know why some transposons have been so successful in one type of species, in one clade, and others in other. Unfortunately, I don't have the answer today for that. What we know is that they have been extremely successful to invade genomes because they have the unique ability to mobilize themselves. And they do that through two mechanisms. The retrotransposons, they have the ability to mobilize by a copy-paste. But basically, transposons are contain genes. The genes are transcribed by polymerase II, like any kind of gene. They are polyadenylated. They go to the cytoplasm. They are translated. They produce the proteins. And the proteins, in the case of retrotransposon, are very important. Are the retrotranscriptase that will recognize the mRNA somehow. They will retrotranscribe, and this cDNA, the DNA copy, will go back to the nucleus and they will be inserted elsewhere in the genome, right? That's why they call as a copy paste. The DNA transposon on the other side, they move as a cut and paste mechanisms. They also are transcribed by polymerase II. The mRNAs are translated and they're producing a protein that calls transposase. And this transposase is very interesting because they have the ability to go back to the nucleus, recognize the cognate transposon, they cut it, and they move elsewhere in the genome. And when they move, the transposon can create many types of mutation. Of course, if they're within an exon, they will disrupt the coding sequence of the gene, produce a loss of function. If they introduce in the intron, they will affect the actin splicing. If they are at the beginning of the gene, they can modify the transcription initiation, premature polymerization. Also, and I will explain a little bit more in details later, but because transposons are heavily targeted by chromatin modification, by chromatin regulation, they can endow the nervine sequences under their own regulation. Of course, also because transposons are transcribed, they typically contain regulatory sequences, and therefore they can affect the activity of genes nearby by the regulatory sequences that contain themselves. And these type of mutations that are typically loss of function produce loss of function alleles are the very same reason that Barbara McClintock discovered them while she was exploring the coloration of the kernel of the maize, and looking at this coloration and accompany this very detailed genetic analysis with microscopy imaging. She could realize that what was happening is that some part of the chromosomes were moving from one chromosome to the other, and there where they discovered, she proposed that this was sequences that were moving from one side to the other, creating variation in activity of genes. And since then, several insertions have been found to be very important for different phenotypes. And here I show you my two favorite examples. One is the transposon insertion, a DNA transposon insertion within a gene called SBI that is involved in starch metabolism in B. And this mutation, which is a loss of function recessive mutation, was the one that was followed by Mendel's, which allowed to derive the loss of inheritance. This is the mutation that produced the wrinkle B. So very important mutation for genetics, for the history of genetics. The second mutation is also DNA transposon, in this case within an intron, but is a gain of function. When the transposon is present, this gene cortex is highly expressed, and they produce a darker coloration of the pepper moth, Piston betularia. And this is exactly this mutation that was underlying the industrial melanism that you probably know from a textbook example of rapid adaptation to human activity. So most of these mutations are typically deleterious, of course, and are therefore highly controlled. The transposon are highly controlled by, in particular, epigenetic mechanism to maintain the transposon in a silent state. And they do that typically by DNA methylation, both in plants and in mammals. We know quite a lot now about how the DNA methylation machinery is depositing the epigenetic marks and what are the impact of these epigenetic marks. Here I will give you a small summary that is important to follow the talk. But I think we know much more that I cannot include in these slides. What I want you to remember is that in plants, DNA methylation can occur in any cytosine. That means that can occur in the cytosine when it has next to a G, this ZG site, when C is followed by whatever nucleotide except a G followed by a G, the CHG, or a cytosine followed by whatever nucleotide that is not a G, followed by whatever nucleotide that is not a G, so CHH. And these three different contexts are maintained by different methyltransferases. CG methylation, here in red, is maintained by MET1, which is the homologue of DNMT1 of mammals. CHG is maintained by CMT3, and CHH is maintained by CMT2. Importantly, all these methyltransferases are orchestrated by a chromatin remodeler called DDM1. DDM1 means decreasing DNA methylation 1 because it was the first gene identified as involved in the methylation of the DNA in Arabidopsis. This is all the system that is involved in maintenance of DNA methylation. And in addition, there is another system that relies in the production of small RNAs. And these small RNAs are produced by a sequence that is already methylated and are processed by several enzymes, including DCLs, RDRs, and AGO, and are targeted to the transposon again by the production of a non-coding RNAs by another RNA polymerase, not POL2, but called POL5. So very important, this RNA-directed DNA methylation pathway requires the activity of two RNA plant-specific polymerase, POL4 and POL5. And this is what we call the RDDM, and it's involved both in the maintenance but de novo methylation. And we know that DNA methylation is important to control transposon because when you mutate, for example, MET1 or DDM1, we lose DNA methylation and some transposon get reactivated. As you can see here, it's the work of the group of Kakutani, where they did southern blot. That's an old-fashioned technique. I will not enter into the detail, but what is important to realize here is that when you do a southern blot against this retrotransposon, copy 93, in the Columbia background, you see that there are typically two bands because there are two locus of this retrotransposon. But if you do that in a DDM1 mutant, you start to see several additional bands indicating that there are many copies of this retrotransposon in the genome, demonstrating that the DDM1 is required to control transposon. Similarly, if you mutate the RDDM machinery, if you mutate the RDDM machinery together with some environmental stress, this also leads to the mobilization of transposon that you can see here. This like a southern blot. Here you have the mutant of the polymerase 5 required for RDDM together with heat stress. So yes, DNA methylation is required to control transposon elements, but also the environment seems to play a role in activating the transposon in some epigenetic mutant in some conditions. So this is what we knew for a long time. Yes, transposons are controlled by DNA methylation, but we didn't know really how much transposition is occurring in nature, right? If you go to a different population of Arabidopsis or whatever individual, how much transposition there are. Second, where are these transposons going? When they create mutation, what is doing randomly, is attacking some genes more than others, and what are the impact of this mutation? That was not very well known. Has some anecdotal examples, but not very systematic knowledge. And the problem is because transposition is typically rare in nature, and most of these transposon insertions are ultimately purged by natural selection, difficulty in their identification when you do comparative genomics. It is the case for any type of mutations. For example, when you want to analyze the mutation rate for production of SNPs, for example, and so on and so forth. And that was a common problem. And population genetics already came out with an experimental system that allows you to study the mutation rate using what they call mutation accumulation lines. This is very simple. So basically, they took, for example, one plant, in this case, an Arabidopsis called 0, which is the reference background of Arabidopsis thaliana. You collect several offsprings, and for each of these offspring, you propagate each of these plants by single seed descent for several generations. And you pick only by single seed descent, so then there is no natural selection, and the mutations are accumulated simply by genetic drift. And at the end of this accumulation of generations, you can sequence these different lines and identify all the mutations that are differing between the offsprings, the 13 generations, and the founder population. I'm doing this several years ago. In this particular is the work of Detlef Weigel. He came out with the mutation rate of Arabidopsis thaliana, which is typically one base substitution per genome per generation, which seems to be continuous during generations. The second is that they found that the mutations are typically even across the genome, so they have similar number of mutations in chromosome arms or centromeres. This is not quite clear right now. It seems that mutations can be a little bit shifted, but overall it's homogeneous. However, they found no transposon insertion. Again, because transposon mobilization is still very rare. So then, if you remember my introduction, I told you that this gene DDN1 is very important to control the transposons. So if you mutate them, we know that transposons get reactivated and they can jump. So then we thought, well, if you take a plant that is a DDN1 mutant, right? Again, we lose DNA methylation, transposons become activated, and we cross with a wild-type plant, giving an F1. Now this mutation of DD1 is in heterozygous state, and we backcross with a cold 0 plant, the wild type, and then we select F2 that are wild type at the DD1 locus. So there is no mutation of DD1, all the epigenetic machinery is there to work. However, this plant came from a parent where we give one generation of opportunity for the transposon to start jumping. And then we asked it, well, maybe transposition was kick-started, and then leads to more transposition. And if we maintain this plant for several generations and we collect the F2 in this case, we can perhaps see the transposons start to accumulate and then start to look at what are the rate of this transposition, where they're going, what are the effect of these mutations. So with that we did, we sequenced more than 100 of these lines. And yes, the answer is there is a lot of transposition in this experimental population that we can call it T accumulation lines, or we also call it epireals because we are also used to study the inheritance of epigenetic changes. What we found in these epireals is that compared with the parental plants wild type, where we found no transposition events, in DD1 we found few new transposon insertions. In this population, we found almost 2,000 new insertions. Most of the insertions were produced by this transposon, copy 93, and two DNA transposon, bundle 21 and SPM3. Very interestingly, most of the insertions were private, meaning that were present in only one individual of this population, right? So one of these line, for example, the line 1 contained some insertions, the line 2 contained a different set of insertions, the line 3 a different set of insertions, and so on and so forth. which by definition means that the transposition occurred during the propagation of the lines, but not in the F1 or in the previous generation. Therefore, because we have so much private insertions, mean that transposition is still ongoing in this population. Okay? And this is very important because now we have a population where transposons become activated endogenously, just because we allow them to lose their epigenetic control and start jumping. And we can start looking where these transposons are going. Here I'm showing you the 5 chromosomes of Biologia italiana. This is the distribution of, in blue, distribution of genes in the 5 chromosomes. Typically, you find the genes distributed on the chromosome arms. This is a typical distribution of plant genomes. This is the transposons that are annotated on the Arabic genome. This is meaning the transposons that have been there forever, right? They accumulated for millions and millions of years. And then you have the transposon insertions that were present in this experimental population. And you can see here that for copy93, these new insertions are typically occurring in the chromosome arms. SPN3 seems to be almost everywhere, and this bundle 21 also seems to be particularly in the chromosome arms overall. We wanted to look that in a little bit more of detail. So what we did is to measure the density of new transposon insertion of the different transposon in genes in blue, intergenic region or transposable elements, and this we compare with expected distribution. And what you can see is that this retrotransposon copy93 preferentially insert within genes, and this is also the case for bundle 21, the DNA transposon. Let's go to a little bit more of detail. When we look what are the density of this new transposon insertion in the 5' UTR, exon, intron, or 3' UTR. What we see, and this plot is maybe it's difficult. You can see in gray is what you will expect in random, and in the different colors you have the distribution of insertions for different transposons. Bundle 21, this yellow, kind of yes, pale orange, preferentially integrate around the TSS of genes. Very strong integration preference for TSS of genes. Copy93 is very interesting because it's almost always present in the exon of genes. This transposon is going where it will produce a bit more harmful. SPN3 seems to go everywhere. And we wonder why these transposons were having different integration preference. We thought that was maybe because there are some DNA motifs that were associated with the preference. We found no sequence association. And then we thought, well, maybe it's the chromatin. They have some chromatin that somehow is allowing or attracting the transposon there. And to test that, what we did is to look at the density of transposon insertion among the different chromatin states that have been defined previously for Arabias italiana. And this is spanning the active promoters, active genes, intergenic, inactive genes, intergenic transposon, and transposons. And we compare what we found with a random expectation. As you can see here, panel 21 has a very strong preference for active promoters. What does it mean? That, well, it's a little bit trivial. This bundle 21 goes preferentially where the chromatin is more open. This is how we can know that, because here we have a plot where we look at the DNA's hypersensitivity or at the sites that this transposon typically go. The DNA's hypersensitivity is a way to measure if the chromatin is open or closed. And therefore, what it's telling us is bundle 21 goes where the chromatin is open. It's just looking at an open piece of DNA in the chromatin, no nucleosomes, and it's inserting there. And this is very reminiscent of the, for example, TN5 that is used for attack-sec. Well, this bundle 21 is a DNA transposon that preferentially go to open chromatin. SPN3, on the other side, they preferentially go to inactive genes, and these inactive genes were enriched with the histone modification H3K27 trimethyl, which is deposited by PRC2. We don't really understand why SPN3 is going here, but still it's very intriguing. The retrotransposon copy93 also go to inactive genes, but a different kind of inactive genes. One that contains the histone variant HUA-CIT. Yeah, this histone variant, you know that the nucleosomes are made an octamer, having 2H3, 2H4, 2H2A, and 2H2B. And this H2A histone, they have the canonical, the typical that is present in most nucleosomes, and variants that are associated with some specific chromatin features, let's say. In particular, the HOS histone variant is present in genes that are typically responsive to the environment. We don't really know why. Perhaps this HOS is a histone that maintains the chromatin in a closed state, but they can be remodeled very easily. So then it's typically deposited in genes that are typically in a silent state, but they need to be activated very fast in response, for example, to an environmental threat. So when we look at the gene ontology of the genes that were targeted by this transposon, as you see, Vandal 21, we found no gene ontology overrepresentation neither for SPN3, but copy identity insertions were very strongly overrepresented in genes, again, response to environmental response. So this was very interesting, but so far this is correlation is not causation. So we wanted to test if or how this HOS histone variant is really playing a role in the targeting of this retrotransposon. So to do that, what we did is to take out one of these lines where transposons are very active, and we cross with the wild type plants. We back cross, and then we cross to keep the control, or we cross with a mutant, a double mutant that is missing most of the HOS histone variant. So these plants are they have very low level of HOS. So we back cross and we select the lines that lack the HOS, and then we collect thousands of offsprings of these lines, and we sequence all together using a way to specifically target the Illumina sequencing on the extremity of the transposons. In that way, we can get very high sensitivity in the detection of transposon insertions. And what we found is that when you mutate the HOS the WMUTA911, we have a much lower number of insertions compared to the controls, already indicating that the histone variant HOS is involved in the activity of this retrotransposon. Second, we lose in the mutant, we lose all the presence for this retrotransposon to integrate environmental responsive genes, indicating or providing very strong evidence that HOS is directly involved in guiding this retrotransposon toward environmental responsive genes. And what does it mean that this retrotransposon preferentially go to immune responsive genes? Well, we can see it in the other way, is that it's not going to other type of genes, in particular essential genes. These genes that, of course, if you mutate, are lethal. And indeed, when you mutate the histone variant HOS, when you don't have HOS, now this copy identity cannot distinguish essential genes or not, and now goes much more often to essential genes than in the control process. So with this, this is indicating that HOS contributes to minimize the potentially lethal consequence of the retrotransposon copy93, and also that HOS likely also guide the integration of other retrotransposon. I'm not showing the result, but we did similar experiment with other retrotransposon, and by analyzing data on rice, and we found that these copy elements has a very strong preference for HOS in most cases. So with this, I would like to summarize the first part of the seminar so far, that I show you that insertion mutations are not random because they have a very strong chromatin-based T integration preferences. I also show you that chromatin plays an essential role in guiding the transposon integration, and that HOS guides transposon retrotransposon TY1 the copia type toward environmental responsive genes and away from essential genes. And I think that this is very important because it's challenging our assumption that the evolutionary process are solely fueled by random mutations, that they typically arouse a slow rate in a clockwise manner. When you have a burst of transposition, these transposons are producing much more mutations. They are not random. They are going to very preferential positions, and when they mutate, the genes are typically producing large effect mutations. That's okay because we have this in the lab, but happen in nature. What is the contribution of transposon mutation to within species variation? So what we wanted to address this, we took advantage of, again, Arabius Italiana, because there are several hundred, even thousands of individuals that have been collected around the globe. This is probably you already know, through the 1001 Genomes Project, led by Dedef Weigel and Magnus Nobra Schoecker. This data, the whole genome sequencing based in Illumina short reads, were already available, and we get all this data and we develop the informatic pipelines to detect the transposon insertions. And we did that, and we found more than 20,000 of new polymorphic T insertions across the more than 1,000 accessions that we analyzed. And that's very important. It was the work of Pierre Baduel, a postdoc that worked with me some years ago. And what he found is that indeed, in Arabidopsis, if you take any pair of accessions, you have several dozen or hundred of polymorphic T insertions. So yes, in nature there are a lot of transposon mobilization. Because we have so many accessions, right, ecotype of Arabidopsis, more than 1,000, we can do very detailed analysis questions to the data. And in particular, we can, for example, take accessions that are closely related, that diverge by a few hundred of SNPs. Basically, they are a few hundred years old of divergence. And we can count the number of transposable element insertion polymorphisms, or TIP, between these any two accessions. And this gives us a linear regression. The more divergent you are, the accessions, the more number of insertion polymorphisms they have. But more importantly, allow us to make a regression and calculate what is the transposition rate in nature. And this transposition rate is around 0.08 new T insertions per genome per generation, which doesn't mean that much, but if you compare with the mutation rate, natural mutation rate, which in look at between accessions is around one mutation every 4 generations. So we are in the same order of magnitude. So in nature, transposition is probably a very important contributor to the major effect mutations, which is typically neglected. So that's a very important message, I think. Second thing, we took each of the accessions of the 1,000 and we count how many recent transposition they have, each of these genomes. And when I say recent, it's insertions that are 1,000 years old. We date all of these insertions. And you can see there are some accessions that have large number of insertions. They have around 80 new insertions in the last 1,000 years. And we have some accessions that contain very few new transposon insertions. And this looks very much as a quantitative trait, right? And I told you at the beginning that in my PhD, I was working with mapping QTLs. And as soon as you see a quantitative trait, you want to do a QTL mapping, right? And this is what we did. We used a genome-wide session study, but using the transposition rate that we calculate by this whole genome sequencing analysis as a trait. I will not give you, I hope that you all know what a GWAS is, but basically try to find the association between the presence of a mutation and a given trait. And each of these dots here represent a different SNP, and in the y-axis is the p-value of the association between the trait and the SNPs. And what you see is that typically most of the SNPs are not associated with a variable transposition rate, but there is a single peak, very strong single peak on the right arm of chromosome 2 that was spanning this gene NRPE1. And this NRPE1, when we saw it, we were extremely excited because this encodes for the measure subunit of the polymerase 5, the polymerase that I mentioned in the introduction that is involved in the RDTM machinery to silence transposon through small RNAs. When this gene is mutated, typically you have more than 200% increase in transposition rate. So yes, in nature, transposition rate is controlled by genetic means. So this I will skip, and I will also show you that we can not only find association with genetic mutations by, for example, mutation in NRP1, but also when we try to find association between the environment where these plants were growing and the transposition rate. And here I show you the world map. Each of the dots represents a different accession of Arabidopsis, and the color of the dot is the level of this transposition activity. The reddish, the more transposition mobilization. And as you can see, it looks like in Mediterranean accessions tend to have more transposition rate than the one that came from the north latitudes. And this is associated, for example, among other variables, with precipitation seasonality. So yes, indeed, it looks like in nature, transmission rate is not only controlled by genetic means, but also by the environment. So the environment is modulating the transmission rate. Okay, so far so good. Now what we look is, we have all the transposition insertion. We can do a very similar analysis that we did with the mutation accumulation lines in the lab to look where the transposition are going. Long story short, these new insertions that we see in nature are very similar, the distribution are very similar to what we obtained experimentally. Basically, these copy and retrotransposons are going to genes, these bundle or mu-DR elements are going to the TSS of genes, and so on and so forth. What was very striking is that typically these insertions are rare. You have very few mutations per gene, but some genes we found that were hit by transposition more than 3 independent times, meaning that you have one accession that came from India that have an insertion in a gene A, and the same gene is also mutated in an accession that came from France, and the same gene is mutated in another accession that came from Germany, independently. So these genes seems to have been common targets of retrotransposition, or transposition in general. And indeed, there are at least 500 of these type of genes that are typically recurrently visited by transposon insertions. And the winner, the gene that contained the highest number of independent transposon insertions, was a gene known as FLC, flowering loci, which is a very well-known gene in plants, particularly Arabidopsis, because it's a negative regulator of flowering time. When FLC is expressed, plants cannot flower, are not licensed to flower, but when FLC is downregulated, and in particular FLC is downregulated by cold treatment, cold vernalization, FLC goes down, silence, and allow the plants to flower. What we found is 16 independent transposon insertion in the first intron of FLC. They have different type of transposon. They have many retrotransposon insertions that we found in the first intron, but also some DNA transposons and gypsy retrotransposon also. And typically, these insertions were associated with a reduction in flowering time. So we believe that these recurrent visits is suggesting a very important role of transposon insertions to local adaptation, allowing plants to flower early in the absence of vernalization. And that's why there are so recurrently found in nature. This gene, is so recurrently found in nature as being mutated. But one insertion in particular caught our attention because, I'm sorry, I will go back, was this particular insertion, this BIB5788 retrotransposon that was present in this HC0 plant. And we caught our attention, I don't have to hear the reason, because the insertion was present there, but the plants were late flowering. And I have it here indeed. When the plants are grown in controlled conditions, we can keep the plants more than 100 days and they will not flower. However, if you vernalize the plants for 8 weeks, these plants flower early, indicating that although they have a 5 kV piece of retrotransposon within the intron, this gene is fully active. This retrotransposon BIB78 is very interesting and is well known also retrotransposon in Arabidopsis because it's reactivated by heat stress. If you treat the plant with heat stress and you measure, for example, the retrotransposon expression by QRT-PCR, this retrotransposon is highly activated, and it was discovered by the group of by Hidetaka Ito, by Jerzy Paskowski in the early 2010. So then we wonder, well, maybe this retrotransposon insertion in FLC is affecting the regulation of FLC in response to a heat stress. And what we did is to treat this HC0 accession with one heat stress at the 2 weeks old plants, or 2 consecutive heat stress, or 3 consecutive heat stress. What we found is that as soon as you heat stress these plants start to flower early. If you heat stress only once, this will lead to low penetrance of the phenotype. So only some of these plants will flower. If you heat stress twice, most of the plants will flower, and if you hit the stress 3 times, they will all flower early. So therefore, yes, now the presence of this transposon is associated with a quantitative response with flowering time. So we wanted to understand this more in detail at the molecular level. So we did RNA-seq, and you can see here this is in control conditions. When the plants are in control conditions, this FLC is expressed nicely. This here, we are showing you the number of reads that are covering the junction of the exon 1 and exon 2. So the gene is nicely expressed. But when you subject to heat stress, now not only you have a very strong reduction of FLC, you have also a strong reactivation of the retrotransposon, consistent with this activity of the ability of the retrotransposon to the activity of this retrotransposon to be reactivated by heat stress. But also we found that all the first intron is covered by reads, indicating that there is all the first intron is being not skipped in the splicing, it's being kept in the transcript, producing a non-coding isoform of FLC. So why this heat stress is affecting the splicing of FLC, the sense orientation, and we thought that, well, maybe this may be related with the epigenetic state of this retrotransposon. So we did bisulfite sequencing, and here I'm showing you the methylation on CG, CAG, and CHH in the control condition on response to heat stress. And what we found is that in response to heat stress, the promoter of this retrotransposon, copy78, becomes hypomethylated in association with the reactivation of the retrotransposon. But of course, again, this is an association. We don't know if the DNA methylation is really controlling the splicing of FLC. So to test that, what we did is to mutate DDN1, this gene that you now know that is involved in methylation of transposons. So when you mutate DDN1, you lose methylation. And we did that by CRISPR, and indeed here you can have the quantification of the methylation of this retrotransposon in the wild type on the DDN1 mutants. And sure enough, as soon as you reduce the methylation, now the plants flower early. This is 2 independent lines, CRISPR lines, and you can see that the plants start to flower very early compared to the wild type plants. And this is indicating that the DNA methylation is required for the normal splicing of the gene, as we can quantify in these plots. So what we did is to did a qPCR, put in primers that are in the first exon and the fourth exon, or in the second exon and the intron. And as you can see is that in the DD1 mutant, we have a reduction of FLC expression and a very strong increase in the production of the non-coding isoform. How this DNA methylation is affecting the splicing of these intronic transposons? Well, work of the group of Tetsushi Kakutani in Japan find out that splicing of heterochromatic sequences depend on IBM2. This IBM2 is an RNA binding protein. We don't know really how it works, but we know that if you we have heterochromatic sequences, IBM2 is helping to mask this part of the transcript to be to allow the splicing of all the intronic heterochromatic sequence. And if you mutate the IBM2, this produces missplicing of the or premature termination of the transcript. So what we did is to test the involvement of IBM2 in the regulation of FLC in this H0 accession. We introgress the IBM2 mutation H0, and sure enough, when you mutate IBM2, now H0 plants flower early. And here, what I'm showing you is that this doesn't affect DNA methylation, so indicating that IBM2 is downstream of DNA methylation, and that the FLC is very strongly downregulated in the IBM2 mutant, and now we have an upregulation of the non-coding isoform intron retention of FLC. So again, IBM2 is required for the methylation-mediated regulation of FLC. Okay, but how we interpret that? So we have a transposon insertion that seems to be affecting the regulation of FLC in response to heat stress. So we say, well, perfect. This may be related with warm environments. We say, well, we should go to the place where this accession was initially collected and found them distributed across the all the region because they share the same climate. So we went to the place where this collection, where this plant was collected. Was very lucky because it's HC0 that came from southern France. We went to exactly the same place where this plant was initially collected in the 50s, in the city of Argentat. We collect dozens of accessions of Arabia of this plant. We genotype for the presence of this transposon insertion FLC, but we found no presence of the insertion. However, in the vicinity of this area, we found two places, and nearby the city of Turin and nearby the city of Arginac, where this transposon insertion was present at intermediate frequencies. And what they have in common, these two places, is one, Turin was on the railway. These plants were collected in the railway, and in Arginac, these plants were collected around around the field where there were staple crops. As you probably know, in Europe, we are not very keen to use herbicides, so therefore what they are using is putting herbicides on the sides of the fields to avoid invasion of plants. And these plants containing these transposon insertions were typically found in these type of places or in the railways. And these two places are typically subjected to very strong herbicide treatment. So that was very intriguing, and what we did is to try to get more data. We have a collaborator that have collected hundreds of Arabiaceae plants from the south of France, and they have identified where these plants came from. And what we found is that here in red the population that contained insertion, in blue the one that lacked insertion. We found plenty of plants that contained insertion. And this is plants that contain insertion were typically associated again with railway, field margins, backyards, or silo sites, reinforcing our observation that this transposon insertion was more often located in places where herbicide was recurrently used. If this is the case, then the insertion may be playing a role in the adaptation to this herbicide treatment, and this should have a footprint on the genomic scale. One way to see footprint of adaptive or positive selection is to look at the strength of the length of the haplotypes. And what we did that using population genomic approaches. What we found is that when you have the insertion, the plants that contain the insertion have much longer haplotype length than those that are lacking insertion, indicating that are under probably current positive selection, and therefore recombination was not. There is not enough time for recombination to reduce the size of the haplotype. So yes, this insertion associated with herbicide and has signatures of positive selection. So when this is the case, then these plants should be responsive to maybe an herbicide, right? And what we did is to grow these HC0 plants in controlled conditions, or during several weeks of fertilization as a control, or we treat with a sublethal dose of glufosinate, which is an basta, is an herbicide, and we count the percentage of plants that flower early. And sure enough, we found that the plants that were grown under sublethal dose of glufosinate, half of them at least flower early. We do it at the molecular level, and we found that this glufosinate is inducing the expression of the retrotransposon BIB78 and associated with a reduction in FLC expression, and again the production of this non-coding isoform FLC containing the intron. So what we believe is happening here is that we have an insertion in a gene that is very important for local adaptation that was selected not for escaping the heat, but probably by escaping the herbicide treatment. So you can imagine that when you have a herbicide treatment, that in this case are not typically complete, there are patches where are more or less affected, the plants that contain this transposon insertion will induce flowering very, very fast and try to produce seeds before dying, providing escape mechanisms to avoid the lethal effect of the weed killers. Okay, so with this I will finish the second summary. What I showed you is that transposon insertion are very often or very common in nature, and these insertions are associated, or the activity of transposon in nature associated with variation in epigenetic pathways and also by the environment. I'm going to show you that the mobilization generates major effect alleles that are associated with local adaptation. And I show you one very interesting example of an internal prototransposon insertion within the gene FLC that provides a DNA methylation mediated regulation, which depends on the diachromatic factor binding factor IDM2. And lastly, I show you that the retrotransposition within FLC enables plants to cycle faster in response to environmental threats, particularly herbicides. And I would like to conclude saying that T mobilization is a major generator of large effect mutations that are finally modulated by the genetic and environmental factors. And the message, if you want, that I want to pass today is that I think the transposon, the mobilization, are making them essential genomic players in the rescue or the demise of native populations in response of very strong and drastic environmental changes. So I think that with this I will finish the result part and start with acknowledgement, the most important part of the talk. I would like to thank everybody in the lab. We have a small but very active lab in Paris-Saclay. Very strong thanks to Basile Leduc, he's a technician in the lab and now also doing a PhD under my supervision. He's very, very motivated. And also show you many results of Pierre Baduel. And that, well, now he opened his lab in Paris too. And also show you some results of Mathieu Rancheval, that he's doing a PhD in Switzerland. Did the master with me. I would like to thank the collaborators, Freddy Barnesh, Nicolas Boucher, Vincent Collot, special thanks, because we work a lot together for the last years. Many of the work I show you today were in collaboration with him. I'd like to thank Ezequiel Petrillo. We have ongoing collaborations. Tetsushi Kakutani from Tokyo, and of course all the agency grants that are paying the bill, and you for your attention, of course.
Thank you very much, Leandro.
Thank you very, very much. Wonderful talk, Leon.
Thank you.
I will open the question. I will tell the students that you can leave your comments and questions in the chat, or you can talk directly to Leandro. I guess Magda has a lot of questions as I know her. So Magda, maybe you can start.
Okay, I will start though. Leon, my first question is regarding, at the very beginning, you mentioned that copia 93, I think, prefers exons. And you know better than me that during the last time we have noticed that gene body methylation induces gene expression. Have you associated something regarding the expression of those target genes for copia 93 and the expression level?
Yeah, very, very interesting question. Yeah, indeed. Yeah, CG methylation in genes is very enigmatic, because there is a significant, it's weak, still the correlation between the expression and gene body methylation. So we don't know what gene body methylation is doing. No idea. Nobody knows. It's probably, it's not about your question, but I think it's an interesting topic. It's probably a leftover or a byproduct of other mechanisms that are leaving the CG methylation there. We don't know. So the genes that are preferentially targeted by the retrotransposon copia typically do not have CG methylation. Do not have, because are lowly expressed. The genes that are highly expressed and are typically CG body methylated are not targeted by the retrotransposon. I think that this is just a evolution of the adaptation of the transposon to try to avoid to go to these genes that are essential, right? If you are highly expressed, you typically have CG methylation and you are essential gene. And these retrotransposons have evolved the way to recognize, okay, this is a gene that is housekeeping, is highly expressed, and this is perhaps very dangerous. You know, if they go there, they will be rapidly counter-selected. So then have been adapted to target the environmental responsive genes. Are typically, as you said, are lowly expressed. Yes, are going to lowly expressed genes. We can speculate there why. And I don't know. I think that, yeah, I think that maybe it's a safe harbor to go to these type of things. The tryptophan that goes to, have tryptophan that go to transposons, typically, right? But these are for the tryptophan itself, it's very dangerous because they go there and you get in a heterochromatic ocean, and then you heterochromatize very rapidly, and then you get silence. If you go to a gene that is not heterochromatic, but almost, then it's a safe harbor. You can go there. Time to time, you will be expressed, but you won't be silenced immediately.
Claro.
Maybe.
Another question. That's something that I missed actually. You mentioned the 0.08 transposable element insertion per generation. What do you mean with generation? I missed that.
Yeah, I mean years, basically. Yeah. Assuming that databases produce one generation per year, right?
Okay.
Which is an annual plant. It's not completely true. It's known that you may have several. Sometimes you can have several generations per year of Arabidopsis, and sometimes you can have less than one generation per year because you have a seed bank that is maintained, and then maybe germinate several years after.
And you are looking only at those insertions that are, in the germinal line. Yeah? You have 1,000 other ones that you will never see.
Yeah. They should be young, because what is the problem? If they are old, you give already enough time to natural selection to remove them, to shift the distribution, to remove the ones that are very deleterious, to fix the ones that are adaptive, and then you get a completely distorted view. So then we want to take the one that are very recent because it's the most closest to the actual rate, right? You hear me? Okay.
Perfecto. And my last comment is regarding FLC. The insertions that you mentioned for this copia 78, I don't remember the number, are all exactly the same. They are.
No, they're all different. Look.
They are all different.
16. Do we have? Yeah. Let me go back.
Okay.
Yeah. No.
Are independent insertion.
Voila. That's what I tried to say.
Okay.
These are 16 independent insertions. One, for example, this one is the one I just mentioned to you, was present in this plant that came from the south of France. And then the other ones, I don't know, but that came from...
No. But that one especially, the first one, is a single event, and you have checked that. I mean, all the experiments that you have done lately with the other plants that you have collected in the railways, and it's, what do you assume? It's exactly the same insertion?
Yes, they have exactly the same insertion. Basically, what we have is we make a genotype in PCR to detect that exact insertion.
Okay.
Yeah, it's exactly the same. Well, that's a good question indeed. It's a good question. I assume that it's the same because they have the same, the same band, that may be independent. But
have you? That's my exactly. That's what. But you have sequenced the flanking regions.
No, I haven't sequenced. That's a good question. That would be extremely. I think that would be extremely unlikely to have. Maybe if that happened, that would be pretty cool. That would be pretty cool.
Because I mean, if you have so many insertions in that intron in different positions, why you assume that in that position you couldn't have, more than one go back and forth?
Yeah.
Okay. So you haven't checked the flanking sites?
I haven't checked. It's good that you are not reviewer of that paper.
Okay. Is there any other question around, Igor?
I have two questions, and Mafros also had one question. So first I will go with my questions. Leandro, you showed that the state of chromatin condensation actually might allow more or less the insertion of these transposable elements. Do you know if there any difference between plants that are, for example, that has holocentric chromosomes? Because then you don't have like one single centromere, and centromeres and telomeres are supposed to have more condensed chromatin regions. So you have these differences, these plants that are holocentric. So do you know whether there's difference in those plants?
Yeah, Igor, you are going to a very hot topic here. Very hot. So, the short answer is no. We don't know. We have no idea what happened with the holocentric. I think that you have probably a colleague from Brazil that is working on this holocentric.
Andre Marques.
And they're doing a beautiful work, right? And they found that, if I remember well, that they have these centromeres that are intermingled with one retrotransposons also. So, so this is very exciting. But yes, but I don't know that. We don't know that. So what we know, and this is what we are doing currently, we have. That's very, very, very interesting. What I find interesting, I mentioned copy 93 is going to genes, no questions. It's going HSZ. This done by our lab, different labs. Yeah, this is that. A colleague, Tetsushi Kakutani, several years ago, 2013, he found that the homologue of copy 93 in Arabiaceae lirata, which is the closest species, this transposons called TAL1. TAL1 goes to centromeres. The transposon is exactly the same, except some part of the integrase that varies. And now the transposon goes to the centromeres. It's the only copy that we know that goes to centromeres. And now we are studying quite actively this because we are starting to realize that there is a very coevolution between the centromeres and the retrotransposon that goes there. The coevolution, the meaning that maybe the insertion of the retrotransposons are affecting the homogenization of these centromeric repeats, and sometimes the retrotransposon can even become the centromere themselves. And there starting to be some reports of some some information that look like there may be several species will have retrotransposon centromeres without centromeric repeats, right? Without satellite repeats. So, Igor, I'm doing a long derivation here, but it will be maybe very possible that the retrotransposon in particular, contributing to the creation of holocentric or distributing centromeres. That would be very, very interesting to assess, to look at. I think that now it's becoming, and you probably know, making whole genome assemblies. As you know, has Ian Anderson is doing a beautiful work on Arabiaceae italiana, but now we know it's Brassicacia, and yes, getting to the bottom of these centromeres. Now it's starting to be more possible. So these holocentric genomes that has a very complex genomes, long, now it's starting to be possible. So it will be very nice to see your advances on that front.
Okay, perfect. And my last question is, I know nothing about transposable elements, but I know that some plants, they have, like, most of their genome is actually made of transposable elements, like maize, if I'm not mistaken, it's like 70%. Do you think, or have you observed something, or in the literature, that these plants, they have like the mechanism that you're showing with this like environmental influence and these mechanisms for adaptation tend to be more important or more frequent in those plants whose genome is more built with transposable elements, or there's no correlation at all?
We don't know, Igor. We don't know. We don't know for the simple reason that most of the experiments I show you we are to do that. We need to experimentally mobilize them and look at them and so on and so forth. And this, you need to have a tractable system. We know quite a bit on maize, though. we could know quite a bit because transposon, as you said, is 80% of the maize genome is transposons, where Barona Clinton discovered. So there are quite a bit of work, and they have plenty of DNA transposons. And for example, MuDR is well known in MuDR type of transposon on maize is going to the TSS of genes. So this is clear. Copia may go to more achromatic regions also, and gypsy retrotransposon typically in the heterochromatic or heterochromatic sequences are accumulated. But I don't know. It will be very interesting to work on that, to look at that.
Okay, perfect. Thank you, Leandro. There's one question from one of our students here in the chat. She thanks you for the nice presentation, and she's wondering whether there are many negative impacts of the transposable elements on the genome, and whether there are strategies to control this negative activity.
Yeah. So indeed, it's a very interesting question. I just pass over that. Why? Because it looks like being an adaptive insertion is more sexy than a deleterious mutation, right? But our calculation means that or indicate that the vast majority of the transposon insertion are either deleterious or neutral. That's the this is the rule. Most insertions are or deleterious or neutral. This depends on the family. We don't have still a real good quantification, experimental quantification. We are trying to do that in the lab. Experimental quantification, what is the fitness effect of each insertion of each different type of transposon. But it's clear that the most mutations are either neutral or deleterious, right? Which makes sense, right? Because it's always easier to break a car than to fix it.
Yeah.
And there was another part of the question I don't remember.
Whether there are any strategies to control these negative effects or activity.
Yeah. So in the term of biotechnological way, I have no idea. I have no idea. In the sense of more in the evolutionary perspective, I think yes, heterochromatic silencing is one way, and it's nice because I have just the screen, the slide with the retrotransposon there, and I told you that these insertions are intronic. If you demethylate them, if they are not methylated, they will affect the activity of the genes. And why? Because they contain the regulatory sequences and they are messing up the splicing. So when they become methylated heterochromatin, then they're recognized and they are spliced out. And are spliced out correctly. So yes,
Your explanation is very exciting. You see.
Yeah. Therefore are spliced out. So I think that yes, DNA methylation or DNA methylation, heterochromatic silencing of transposons first are inactivating them to jump, and second, when they jump, maybe reduce their effect in some cases. But it's a double sword. It's a double. Yes, they have two effects. Heterochromatic silencing can also affect the activity of nearby genes. So.
Okay, Leandro. We have a question on YouTube from Professor Michel Vincent. He congratulates you for the nice work. He asks you if there is any specific reason for Pol5 polymorphism in natural population being involved in higher transposable activity, or could we expect other genes involved in epigenetic control modulating the transposable activity?
Very nice. Very nice question indeed. So yeah, NRP1 and NRPD1. So we found NRPE1 mutated. We didn't find NRPD. So that just in the jargon, we found Pol5. They are independent. We have many different alleles of Pol5 mutated. They are not loss of function. There are variants typically on the carboxy terminal domain, the CTD, that are affecting how Pol5 seems to recruit cofactors for their activity. So they are hypomorphic alleles. And this is in Pol5. But we never, so far, I never found mutants on Pol4. And this is a matter of discussion, why Pol 5 is so prone to vary instead of Pol 4. And I don't have the answer, but speculation here is that Pol 4 is required for the production of small RNAs, and the small RNAs has a, if you want, they have a double function. One is to produce the small RNAs that can interfere with the transcripts, like post-transcriptional gene silencing. They can fit the post-transcriptional silencing, and the classical is to fit the transcriptional gene silencing, the RDDM. Whereas Pol 5 only works for on controlling RDDM, on the factor of the PTGS, the post-transcriptional gene silencing. So perhaps affecting both the transcriptional gene silencing and the post-transcriptional gene silencing is very deleterious. But you can tolerate to have a reduced PTGS. This is speculation, and I think it's exciting. Why this differentiation between the mutation in different pathways? Similar, we are. I don't know. Maybe there is another question, or I can continue elaborating a little more. That
you can elaborate a little bit more.
Okay. So I also mentioned the CMT2, CMT3, which are chromomethylases that are involved in the deposition of methylation. CMT3 on CHG methylation, CMT2 on CHH methylation. CMT3 we never found there are not mutants of CMT3 in nature in accessions, but there are null mutants of CMT2. CMT2 has found plenty of mutants of CMT2, indicating that CMT2 is completely dispensable. And CMT2 has a very strong impact in methylation. You lose most CHH methylation. It's very impressive, and plants live okay. You go to the field and you find plants without CMT2, without CHH methylation on transposons, which call to the question what this pathway is doing. Why CMT2 is there? If you can mutate and be okay with that, it's probably you are not okay, right? Because it's not fully okay. And I don't know. I don't have the answer, Igor. I don't have the answer, but it seems that the different pathways still there. Although we know a lot, there are many things to learn, and I think that this epigenetic pathway at this level they may contribute to the flexibility of the genome, right? Allow to breathe the heterochromatic silencing more or less, and this may be useful in some situations. I don't know, maybe during hybridization, maybe during whole genome duplications, maybe I don't know. It's speculation, huh?
Okay, Leandro. There is one question from Dr. Mafros Bazunia. This is more towards biotechnology, whether your research will have some future implications to improve cereal crops or maybe other crops yield in climate change hotspots.
Wow. I don't work in crops. I work in typical Arabiopsis. We are working also in tomato. I didn't present anything on tomato just for a reason of time. But yes, I think that yes. So I think that there are, for example, Etienne Boucher, who is working in Switzerland. He's an expert on transposon element. He's using transposon to generate variability by modulating their activity, by knowing where they're going, and so on and so forth. You may you may control them on your benefit for breeding to improvement. And these are interesting possibilities. I think that we are still, there are kind of transposons still that are very wild, very savage. So I think that we are still far away from controlling them and making them do whatever we want. But if you continue working on that direction, we will manage. And one thing that I want to add on that respect is that, as I show you, the transposons have, there are genomic entities that have evolved to interact with the genome, with their own genomes, with their own genome. So they have evolved very sophisticated ways to recognize themselves, to recognize the type of chromatin, to go to this place but not the other one, to jump in this particular tissue but not in the other tissue. And we have a lot of opportunities for us through understanding the transposon, understanding how we can modify the genomes in a very smart way, because they have been doing that since the beginning of life.
You know, that's very interesting, because one of our students here, Gustavo, he thanks you for the great talk, and he's wondering whether it's possible to use transposable elements to precisely edit genomes.
To do what with the genome?
Do genome editing. I mean,
Yeah. So
because
yeah,
you can go to a
yeah.
place.
So prime editing, you probably are related, you hear about prime editing. It's a different system, right? But people are starting to work on fusion of integrases to integrate pieces of DNA exactly where you want, right? And this is very exciting, right? Imagine you bring the integrase to one place, then you put whatever sequence you want, as soon as they have the correct terminal repeats.
Yeah, that's nice.
And I think that also, and I think that Keith Slotkin, yeah, I think Keith Slotkin is using, in this case, DNA transposase, because the transposase are beautiful, because they have ability to cut the DNA exactly. They have the scissor and the glue at the same time. So yeah.
Okay, thank you, Magda.
I have a question. As there are so many different transposal elements that are able to insert in heterochromatin, I wonder whether you have any clue about how can they reach the heterochromatin indeed.
Yeah.
Because, I mean, we always repeat, heterochromatin is not accessible
but
to RNA bond because the protein cannot get into. But these guys are able to get into the DNA, cut the DNA, insert the transposon in the middle of an heterochromatin. So they must have a chromatin remodulating activity somehow. How can they reach that from the molecular point of view?
That's an open question, Magda. You want to answer for me? I don't have the answer. That's very that's what I said, that they have evolved to, they are very sophisticated interactors of the genome. They know the genome better than us.
By nobody, no, I mean, there is no reports about any chromatin remodeling domains, let's say, or another, because I remember, I mean, the structure of the transposal elements have the transposase or the RNA pole or blah, 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 but I don't remember anything about something that remodels chromatin, you know what I mean?
It's very interesting. It's very interesting, your question. So we are trying to look at that. We are trying to look at that. We are trying to look to what the integrals are interacting, particularly those that are going to heterochromatin.
It might be something
it could be that
hidden. I mean,
they could be that activity interact, for example, with DDN1. DDN1 is a chromatin remodeler involved in heterochromatin remodeling, or other factors. That could be. It also could be that they are jumping in a particular moment of the cell cycle where this is accessible. During S phase.
Exactly.
S phase, you are heterochromatin, but you need to allow the polymerase to go through, right? So, and it's also true that we have this heterochromatin is closed, but if you do attack sec, you access the heterochromatin. No, you get it. We recently implement a protocol to put adenine methyltransferase within nuclei, so then we can methylate all the adenines, and then we can extract the DNA. And then if we have an adenine, we know that was accessible, and then we can sequence the nanopore, and we can read both the methylated adenine and the methylated cytosine, right, at the same time. So then we know, well, this was heterochromatic because was fully methylated the cytosine, but has also adenine methylated, and then we know that was accessible. And what we found is that, so the heterochromatin is accessible. It's less accessible, but it's accessible. But maybe chromatin remodeler, maybe there is something. Yeah.
Great.
Nice.
Okay, I think that's it, Leandro. We don't have any more questions. So I would like to thank you very much for your very, very nice and interesting talk and discussion. So on behalf of the Department of Botany from our university, I thank you very much for your collaboration. And and thanks everybody for attending.
My pleasure. Really, my pleasure. Very nice.
Muchas gracias,
Very
Lean.
interesting discussion.
Un placer.
El placer es mío. Gracias, Magda, por todo lo que hiciste por mí.
Muchas gracias. ¡Mua! Besos, miles.
Adiós.
Besotes.
Bye.
Bye, bye.
Chao.
Thanks.
Ciao,
Thanks.
ciao,
Bye, bye,
everybody.
Paul.